Welcome back to Module 3 of Anatomy and Physiology Systems 1, the Nervous System. Topic 3.2 will cover the central nervous system. If you open your workbooks to Module 3, try to answer as many questions as you can as the mini-lecture progresses. This is Part A of the central nervous system, where the focus is on the brain. The topic of this mini-lecture is the overview of the central nervous system. Because the brain is such an important and substantial component of the nervous system, the central nervous system will be divided into three parts. Part A and B will detail the brain, and Part C will cover CNS protection and the spinal cord. The learning outcomes for Topic 3.2a are: to know the anatomical structures and the function of these regions of the brain, the cerebral hemispheres and lobes, the major gyri, fissures, and sulci. To start with, let's review how the nervous system is organized. The central nervous system consists of the brain and the spinal cord, and is essentially the master controller of the nervous system as a whole. The adult brain contains four regions that we will discuss: the cerebral hemispheres, the diencephalon, the brainstem, and the cerebellum. The remainder of the central nervous system consists of the spinal cord. Grey matter and white matter are uniquely distributed throughout the brain and spinal cord. Grey matter in the CNS consists of short, non-myelinated neurons and neuron cell bodies. White matter is composed of both myelinated and unmyelinated axons. In both the brain and the cerebellum, there is a thin outer layer of grey matter called cortex, surrounding the white matter, deep within the brain and the brainstem. However, in the spinal cord, the opposite occurs, where grey matter is found innermost, surrounded by white matter. Before we go on to explore these regions of the brain in detail, let's first explore the central hollow cavities that lie deep within the brain, called the ventricles. The brain ventricles are continuous with one another and with the central canal of the spinal cord. The hollow chambers are lined by the ependymal cells, one of the types of neuroglia that we have previously discussed. They are filled with cerebrospinal fluid, or CSF. The large paired lateral ventricles are C-shaped chambers that reflect the pattern of cerebral growth and located one in each hemisphere. They consist of anterior, posterior, and an inferior horn each. They are separated by a very thin membrane called the septum pellucidum. Each lateral ventricle is joined to the narrow third ventricle by small channels called intraventricular foramen. The third ventricle is contained deep within the diencephalon and is continuous with the fourth ventricle via the thin cerebral aqueduct, which runs through the center of the midbrain. The fourth ventricle lies anterior to the cerebellum and posterior to the pons and medulla of the brainstem. Inferiorly, the fourth ventricle is continuous with the central canal of the spinal cord. Additionally, there are three little openings within the walls of the fourth ventricle that allow the ventricles to connect with the subarachnoid space surrounding the brain and the spinal cord. The two cerebral hemispheres form the superior part of the brain and account for 83% of the total brain mass. Elevated ridges of tissues are called gyri, and these are separated by shallow grooves in the tissue called sulci. Deeper grooves in the tissue are called fissures, and these separate larger regions of the brain. The more prominent gyri and sulci are important anatomical landmarks. For example, the central sulcus separates the frontal and parietal lobes of the brain. The lateral sulcus separates the temporal lobe from the frontal and parietal lobes. The deeper longitudinal fissure separates the two cerebral hemispheres. The cerebral hemispheres fit snugly inside the skull. Each hemisphere is divided into 5 lobes: frontal, parietal, temporal, occipital, and the insular. The first 4 are named for the cranial bones that overlie them, and the 5th lobe, the insular, is found very deep within the lateral sulcus, covered by portions of the temporal, parietal, and frontal lobes. What's the purpose of all the gyri and sulci? Why is the surface of the brain folded and not smooth? The many convolutions effectively triples the surface area of the brain, where more neurons and cell bodies can be contained in a smaller space. The superficial cerebral cortex, consisting of grey matter, is where our conscious mind is found. This enables us to initiate movement, to become aware of ourselves and our sensations, to communicate, to remember, and to understand. The grey matter consists of neuron cell bodies, dendrites, glia, and blood vessels, but no fiber tracts. Although about 40% of the total brain mass is contained within the cerebral cortex, it is only about 2 to 4 millimeters thick. The cerebral cortex can be divided into 3 functional areas: motor areas, sensory areas, and association areas. Each hemisphere is mainly concerned with the sensory and motor function of the opposite side of the body. However, none of the functional areas of the cortex acts alone, and conscious behavior involves the whole cortex, not just a specific functional region. Firstly, let's take a look at the frontal lobe. The posterior part of the frontal lobe contains the motor areas of the cortex, which control voluntary movement. These include the primary motor cortex, the premotor cortex, Broca's area, and the frontal eye field. The primary motor cortex is found in the precentral gyrus of the frontal lobe, the region of cortex that lies anterior to the central sulcus. Large neurons called pyramidal cells with long axons project to the spinal cord, and these form the corticospinal tracts. The corticospinal tract allows us to consciously control precise voluntary movements of our skeletal muscle, such as kicking a ball or flexing the elbow. The premotor cortex is located just anterior to the precentral gyrus and the primary cortex. This motor region helps plan movements and to sequence basic motor movements into more complex tasks, such as playing a musical instrument or typing. It's the region that helps perfect your golf swing. The premotor cortex is responsible for coordinating the movement of several muscle groups, either at the same time or one after another. Another motor area located in the frontal lobe is the Broca area. This region lies anterior and inferior to the premotor cortex. This area controls speech production. It is a special motor speech area that directs the muscles involved in speech production. It becomes active even as we prepare to speak, and is found in the left hemisphere only. If the Broca's area is damaged, the condition is termed expressive aphasia. The person can understand the conversation and can think the word they want to use in response, but when it comes to producing language, they can't actually say it. The frontal eye field is located anterior to the premotor cortex and superior to the Broca's area. This region of the cortex controls voluntary movement of the eyes. If there is damage to the frontal eye field, the person is unable to look away from the side with the injury. The parietal lobe is concerned with sensation. The primary somatosensory cortex is found in the post-central gyrus of the parietal lobe, just posterior to the central sulcus and primary motor cortex. The primary sensory cortex is responsible for our conscious awareness of sensation. Neurons in this gyrus receive information from sensory receptors in the skin and from proprioceptors in skeletal muscles, joints, and tendons. The neurons then identify which area of the body is stimulated. The somatosensory association cortex lies just posterior to the primary somatosensory cortex in the parietal lobe. Its major role is to integrate the sensory inputs relayed to it from the primary somatosensory cortex, so that the person may have an understanding of the object being felt. For example, if you reach into your pocket and feel a coin, the somatosensory association cortex uses stored memories of the past sensory experiences to perceive the object you feel. If this area is damaged, the person is unable to recognize something by touch. The primary visual cortex is also known as the striate cortex. It is found in the extreme posterior tip of the occipital lobe. This region receives visual information that originates on the retina of the eye, which creates a contralateral map of visual space on the primary visual cortex, similar to the body map of the somatosensory cortex. The visual association area surrounds the primary visual cortex and covers the majority of the occipital lobe. This region uses past visual experiences to interpret stimuli, to allow us to recognize familiar objects, such as a person's face or a type of flower. Each primary auditory cortex is found near the ear, in the superior margin of the temporal lobe, just next to the lateral sulcus. When the hearing receptors of the inner ear are excited, impulses are transmitted to the primary auditory cortex, where they are interpreted as pitch, loudness, or the location of sound. The auditory association area is located slightly inferior and more posterior to the primary auditory cortex. This association area is involved in the perception of the sound stimulus. That is, meaning is added to the type of sound heard, such as whether the sound is a scream, music, or thunder, for example. Deep on the medial aspect of the temporal lobe, the olfactory cortex is located, in a small region called the uncus. Afferent fibers from the smell receptors in the nasal cavity send impulses along the olfactory nerve to the olfactory cortex here. The olfactory cortex provides us with a conscious awareness of different odors of smell. Deep to the temporal lobe is the fifth lobe of the cerebral hemispheres, called the insula. This is where the gustatory cortex is located. This region of the brain is located deep to the lateral sulcus and is involved in perceiving taste stimuli. These two regions, the olfactory and gustatory cortices, are so closely related that we can often taste a horrible smell. All of the association areas that we have considered so far have been tied tightly to one kind of primary motor or primary sensory cortex. However, as you can see in this diagram here, there is a large percentage of the brain, coloured in violet, that consists of complexly connected multimodal association areas. These regions receive input from multiple sensors and send outputs to multiple areas, so generally information flows from the sensory receptors to the primary sensory cortex of the sensory association cortex, to the multimodal association area. This helps us give meaning to the information that we receive, store it in memory, and decide what action to take. Our thoughts, sensations, and emotions become conscious when we can tie them to previous experiences. We can divide these multimodal association areas into three regions: anterior, limbic, and posterior. And we will discuss these three areas in the tutorial sessions. The cerebral hemispheres aren't identical or exactly symmetrical. There are unique abilities known to each hemisphere, which we term lateralisation. The term cerebral dominance actually designates the hemisphere that is dominant for language. The left hemisphere controls language function, maths, and logic. These abilities help us to compose a sentence, memorize a list of activities, and add a series of numbers. The right hemisphere, instead, controls visual spatial skills, intuition, emotion, and artistic and musical skills. While each hemisphere is better than another at certain functions, it is not completely black and white. In reality, and there are individual differences that do occur. This concludes our mini lecture of Part A of the brain, Topic 3.2, the central nervous system.